Hello, Chem 103. Hope everybody's doing okay. Today, we're going to talk about getting ready for the lab this week, which is why we're doing it on Tuesday instead of our normal Thursday. It's important that you kind of know what's going on. This is going to be our most complicated lab so far. There's a lot of things that we're going to do, a lot of different bits to it. So today, I'm going to go through all of that. And when the group A comes in tomorrow, I am going to expect that you have watched this and kind of gone through this material as your pre-lab preparation, because we really won't have time to go through everything in class tomorrow, other than showing you how to use the equipment. So this is to help you be prepared for that. And then for group B, same thing, you just have a little bit more time. You'll be in next week also doing this. The advantage for group B is you'll have a little bit more time to prepare for the lab. The advantage for group A is you have a little bit more time after to do the analysis. So today we're going to go through the pre-lab stuff and all the various parts of this experiment, the biodiesel analysis. And then next week we will go through the calculations. And we're going to do that on Thursday so that by then everybody in the class has all of their data and you can follow the calculations through together. All right, so let's get started. If you go into the assignments folder this week, whoops, sorry, wrong folder. That's just where you're going to submit the stuff. If you go into the activities folder, there's one balancing equations activity to do similar to our other ones. I think this one's fairly straightforward. I don't think you'll have any issues with it. Let me know if something comes up. I'll mention that on next Thursday as well. But the main part is going to be in this biodiesel project folder. So the biodiesel project, as we have talked about, is a long, several weeks long project that we are basically finishing up this week and next week, at least for the lab part. Then you'll have a little bit more time to complete the rest of the project. So the actual analysis, the actual final project is this document right here that says Biodiesel Project Final Analysis. So if we pull that up, hopefully this is loading okay. Yep, there it is. Okay, so this is the final analysis. You will be answering all of these questions as your biodiesel project about your stuff. So several of the questions are kind of more research-based, like what is the purpose of a fuel? Why is this a renewable fuel? And so on. What were the results of the water wash tests, things that you've done or that you can find out. And then the other ones, like number 1, did you produce biodiesel, what we thought we were producing? And then calculating the theoretical mass and the percent yield. These are things we'll do together. We'll do these calculations together. And then the heats of combustion, which is what we're going to measure in lab. So these are all based on the data that you get in lab. And you'll be getting all those data this week or next week, depending on if you're in group A or group B. All right, so for this week, what we're going to focus on is the experimental biodiesel analysis. So this is what we're doing in class, in lab. And so there are several tests that we're going to be doing to your biodiesel. So I'm going to just kind of go through and describe each one and what we're looking for and what they are about. And then we're going to focus most of our time on this last one, the heat of combustion, which is going to be what we spend most of the time on Wednesday. All right, so these tests all concern themselves with characterizing the product that you got and seeing if it is indeed biodiesel, if it's pure, or if it has other like unreacted oil in there or something like that. We'll be able to figure that out. And then we'll also know some things about what it's composed of and how it behaves as a fuel after getting all this stuff. So wash test, you're just mixing the fuel with some water in a flask and you shake it up. And the reason for this is just like you did last week when you did the washing. If the mixture separates pretty quickly and it's a nice clean layer or it's a nice clean line that separates the biodiesel from the water, then we know that that there aren't these surfactants and other types of impurities in there. If you get a really cloudy thing with like a layer of kind of white cloudy mess in between the biodiesel and the water, then there's some impurities in there. The methanol test is a good way to test for unreacted oil because the biodiesel dissolves in methanol, but the original vegetable oil does not. And so you try to dissolve a little bit of your biodiesel in methanol, and then you look for any droplets of undissolved oil still there. And if you see evidence of that oil, that's evidence that the reaction did not completely use up all of the oil that you started with, and that there's some leftover methanol there, leftover oil there. All right, the cloud point is an important data for fuel. That is the temperature below which crystals start to form. And when we say crystals, we're not talking about like nice looking, shards of shiny things. We're really just talking about it kind of clouding up, starting to form solids. And so you can imagine, especially in our climate, if you have a fuel that is going to cloud up around, normal winter temperatures, that's going to be a problem because those solids are going to clog fuel filters. They're going to cause deposits in an engine, things like that. So that's a potential drawback to certain fuels, and certain fuels have lower cloud points than others. So we'll be able to compare what you get by cooling your fuel down and looking for that cloud point with what's reported for other types of fuels. So to measure the cloud point, you're going to have to develop a plan of how you want to do it. But basically, it's going to involve cooling a sample of your biodiesel and then looking for when it starts to cloud up, when it goes from clear to cloudy, which is evidence of some solids starting to form. All right, so you can look, before you come into lab, you can look up a little bit how to measure cloud point, best practices for that kind of thing, and then develop a little plan in your notebook for that. Okay, same with density. Develop a plan to measure the density of your fuel. The density is not so much about if it's pure or not, but you can compare to reported densities for commercially produced biodiesel, and you can see if it's in the same range or if it's way off. So look up how to measure density and get a plan for doing that in lab. It's pretty simple. Essentially, you need the mass of a given volume, and then you divide the mass by the volume. So you measure like a milliliter or 2 milliliters or something, and then take the mass of that amount and then divide those two numbers. And you should do it a couple times, maybe a couple different volumes, just to get consistency, make sure you have consistent numbers. All right, the infrared spectroscopy and GCMS, these are instruments that we would use to measure the biodiesel. These are going to be dependent on time. So we often skip the infrared spectroscopy because we just run out of time and it doesn't tell us that much. What we can do for the GCMS, this measurement actually takes a while, but we can you can prepare samples while you're in lab, and then I can run them on the machine after lab is done and just send you the data later, because each measurement takes like, I don't know, half an hour or something. So it's not really practical to do during the lab, but we can get the samples set up and then run the machine later and just send you the data. So what this is, kind of like the chromatography we did before, but this time it's a big fancy machine, and it passes, it first heats up the sample to turn it into a gas, and then passes it through a really long tube filled with special materials that some chemicals bind to better than others. And that separates the different components of your mixture. And when it comes out the other side, each particle that comes out is essentially weighed by something called a mass spectrometer, where it can use the mass of the thing that comes out to figure out what that molecule is and look it up in a database. So this is a really powerful way to actually identify exactly which molecules are in your sample. And that will be probably the strongest proof of what you've made when it comes out. So we'll definitely try to get that data. We just may not be able to do it during class tomorrow and next Wednesday. Okay. So then the bulk of our time tomorrow is going to be spent measuring the heat of combustion using a bomb calorimeter. So let's talk about that for a second. So first of all, that has its own procedure, which is the third one down here, bomb calorimetry. So make sure when you are getting, all your materials together for lab, also print this one out. You'll need this as well. All right. So what is calorimetry? Well, let me pull up a blank paper here. Calorimetry is measuring energy. So basically, we take a something that is going to change energy. Either it's going to give off energy or get put in energy or whatever. And we measure the change in energy by putting it in some sort of environment where the energy can't escape to the surroundings, and then measuring the temperature before the thing happens and after. And the change in temperature is caused by the movement of energy. So we use this this unit of calories to do that. You've probably heard this term before, calories, but we're going to define it here. 1 calorie is the amount of energy that it takes to raise 1 gram of water 1 degree C. So if you have 1 gram of water, which is also 1 milliliter because the density of water is 1, changes a little with temperature, but it's basically 1. You have that much water. If you raise it from 25 degrees C to 26 degrees C, you've put in 1 calorie of energy. So if you raised it 100 degrees, you put in 100 calories. Or if you had 100 grams of water, so a beaker full or something, and you raise that 1 degree, that would be 100 calories. If you raised it 5 degrees, it would be 500 calories. So basically, we can calculate the heat, which is the transfer of energy, and it's given by this variable Q, is going to be the mass of water times the change in temperature. And there's actually a third term in this called the heat capacity, because if it's not water, there's an additional correction there. But because we've defined water, or we've defined the calorie as the amount of energy it takes to raise 1 gram of water by 1 degree C, then we can find the heat by just taking how much water we've got and then how much it changed in temperature. A couple other things you should know about calories while we're talking about them. This calorie that we're talking about here is not the same as the calories on your food. It's a little bit confusing. Foods use capital C calories, which are actually 1,000 little c calories, and more correctly called a kilocalorie or a kcal. So you may have heard that. Most other countries measure their nutrition in kcals. We're measuring it the same way here, but we use this capital C calorie. So that can be a little bit confusing, of course, because C is a letter that looks the same as a capital or a lowercase. So it's hard to sometimes keep those straight. When we measure in calorimetry, we're measuring little c calories here when we're measuring this Q. When we're measuring foods, and we want to compare to labels, we often have to divide by 1,000 to change that. And we might do a fun food calorimetry experiment later in the semester with one of the to be announced lab days. All right, so that's how we do this, like, theoretically. So then physically in lab, what does this look like? So on Blackboard, there is a picture of a bomb calorimeter. And I'm going to kind of take it apart for you too. This is the thing, what it looks like. It is a heavy steel-walled thing, and it is meant to maintain its volume. So it doesn't get bigger, it doesn't get smaller, it's just a hard steel container. Inside that container, I got a lot of things open here, hold on. Okay, inside that container kind of looks like this. So you can unscrew this outer bit here, and then there's this whole setup inside that holds a little pan in this little hook thing. And I'll go through all of this in lab to show you the setup thing, but I want you to kind of know what the idea is here. So when we do calorimetry, we take this thing called the bomb or the calorimeter. This is now kind of cut open, and we submerge it in water. The reason we submerge it in water is because we're going to ignite or burn the biodiesel that's inside the calorimeter. And we want to capture all of the heat that comes out. We don't want the heat escaping to the air. So we put it underwater, under quite a bit of water. We're going to use 400 milliliters of water, so kind of a big bucket full. And that way, all of the heat that's produced by the burning biodiesel makes its way out into the water where we can measure the change in temperature. And so that's what's going on here. This setup is not exactly the same as ours, but same idea. We have a calorimeter that's underwater. We have an ignition coil that is inside there that is a wire that's connected to wires up here, and it's going to heat up, and that's what's causing the biodiesel to ignite. We don't have heaters on the side, so we don't have to worry about that. And then once the biodiesel ignites, that heat goes out through the calorimeter. It heats up the calorimeter as well, but it goes out to the water. And then we have a thermometer. We're actually using a digital setup that's hooked up to a computer, so we can monitor the change in temperature as that heat kind of filters out into the water. So then we can use the equation, although we're going to adapt it a little bit to make it a little more precise, to figure out how many calories of energy were in that particular sample of biodiesel. A couple other things that make this work properly. If you, as you've probably experienced, if you try to just light something on fire, it often doesn't combust completely. You get a lot of ash and soot and stuff like that. And that's not good for a quantitative method like this, because that's stuff that didn't burn. So in order to have full combustion, we need to make sure there's plenty of oxygen, because combustion is when a fuel reacts with oxygen. So when you burn something in air, there's actually not enough oxygen usually for full combustion, and that's why you get ash and soot. But in the calorimeter, we're actually going to pressurize it with pure oxygen, so that it's in a pure oxygen environment inside there. That way, when it ignites, it has all the oxygen it needs to totally consume the biodiesel. So when you open it back up, you won't see anything, any evidence of combustion at all. It's all turned to carbon dioxide and water vapor. So then we'll use that, and we'll do it a few times so that you get some good data depending on time in lab, to measure, to do a couple of these measurements. So then when we have the data, then we'll have some measure of reliability there, because we'll have some reproducibility, hopefully. Okay, so back to the instructions here for the bomb calorimetry. So these are the specifics of how we do this experiment in lab. We're going to, you're going to work through these steps. I'm going to give you a demo of it at the beginning of lab tomorrow, but I'm going to expect that you've read through this and have at least a sense of what's going on and some of the numbers that you're going to be using here. It's a lot of steps, so I don't expect you to memorize this. I don't expect you to write all of this in your notebook. I just expect you to have read it and have a good sense of what's going on, and to have listened to me kind of talking about it through here. The other thing that we're going to do tomorrow, and this is specifically for number 2 and 3 here, is we have to measure how much biodiesel product you got. So we're going to take it out of the separatory funnel. We'll drain off the bottom layer and then take out the top layer and measure how much of that top layer you've gotten. And then next Thursday, we'll work through some of those calculations. It's not as simple as just subtracting the final mass from the initial mass because the identity has changed. It's no longer the same thing. It doesn't weigh the same amount as a molecule. So those characteristics have changed, and so the calculation is a little bit different. Again, it's not something that I'm expecting you to do on your own. We're going to work through it together, and you're going to put in your numbers and have your data. I think you can interpret the data fine on your own, but doing the actual calculations gets a little tricky. So we'll do that part together. All right, so this is for pre-lab. So please make sure that you, of course, are dressed appropriately for lab. Check your status being cleared for Harper. Make sure that you are indeed cleared so that you can come and show a green pass if you have to when you get to campus tomorrow. And then make sure you've read through the instructions, specifically the experimental analysis here, and you've looked up and thought a little bit about your plan for measuring the cloud point and the density. And then make sure you have the instructions for the bomb calorimetry as well. I would really encourage you to have printouts for these. If that's something that you can't do at home, if you come to campus a little bit early, all the computer labs are open now. The Mega Lab in Y, which is the building right next door, you can just go in there and print using your student ID so that you'll have copies of this to work on during the lab. And then, of course, bring your notebook also. All right, so if anything comes up between now and tomorrow for Group A, please let me know. So you're ready. For Group B, you've got a little bit more time to think about it, but I think this will be fine. It's a lot, so I wanted to make sure that we talk a little bit about it before we get started in lab. All right, thanks everybody, and I'll see half you tomorrow and the rest of you next week. I'm not going to meet online on Tuesday morning this week, so it'll be next week that we do the analysis and the calculations together. All right, thanks everybody. Bye.